So we kind of talked about these questions already. What are the roles of ATP in muscle contraction? So what are the two uses of ATP in muscle contraction? The first is to what? To break cross bridges. So the first role is ATP can bind to the myosin head to break cross bridges. That's kind of like the last step of cross bridge cycling. What else does ATP do? That would be for relaxation. But for contraction, what's the other role of ATP? What's the main action in the cross bridge cycling? What do we call that action? The power stroke. So you can say the other role of ATP during muscle contraction is it energizes the myosin head so that the power stroke can occur. ATP energizes the myosin head so that the power stroke can occur. The power stroke is the main action allowing for cross-bridge cycling. And then the second role of ATP was it breaks a cross-bridge so that we can repeat that cycle again, so that we can get full and the desired amount of muscle contraction. So those are the two roles of ATP in contraction. And then Ariana said one role of ATP in muscle relaxation. What's that? Yep, it pumps, it powers the calcium pump so that you can move calcium back into the sarcoplasmic reticulum and get it away from the thin filament. Because calcium is needed for muscle contraction, if we want muscle relaxation, which is the opposite process, we need to get rid of the calcium. And we do it by pumping it back using ATP, and we pump it back into the sarcoplasmic reticulum. What's the other use of ATP in muscle relaxation? It's also a shared feature in muscle contraction. We use ATP to do what? To break the cross bridges so the muscle can relax. Or otherwise you'll be stuck in the rigor state. The third question is, how do we generate ATP? Well, during our cell respiration lectures, we talked about a variety of ways we can generate ATP, and we'll review some of them here. So muscle metabolism, both muscle contraction and muscle relaxation uses a lot of ATP. How do we provide that to the muscle? Well, for some short-term ATP supplies, in the sarcoplasm of muscles, there's some ATP, not a lot, but some ATP just readily available that the muscles can use for muscle contraction and muscle relaxation. And we refer to that as intracellular stores of ATP. It's ATP inside the cell. So there's not a lot of it, that's why it's under short-term supply of ATP. We can also use creatine phosphate to make ATP for our muscles. So creatine phosphate, we can abbreviate it CP. And if we have ADP, what we can do is transfer the phosphate from creatine phosphate onto ADP to make just creatine and ATP. So we took the phosphate off of creatine phosphate, we transferred it onto ADP to make ATP. And this will give your muscle a little extra ATP when needed. Glycolysis makes how many ATP? 2. So thus it can make a little ATP and provide a short-term supply of ATP to your muscle. Now, long-term ATP supply would be through this whole entire process, aerobic cellular respiration, where we can make a lot more, like a total of 32 ATP if we break down glucose. Or if you break down fatty acids or triglycerides, we can make hundreds of ATP. And because we can make more ATP by using these energy sources, it provides a longer supply of ATP for muscle contraction and muscle relaxation. So we can get glucose from glycogen stores of the muscle, as well as glycogen stores from the liver. And then we can also feed triglycerides and that glucose from the blood into our muscles to make a lot of ATP. Let's talk about botulinum toxin, which is a neurotoxin that could be deadly and make you paralyzed, or it could have some clinical or even cosmetic uses for your body, which we'll look at shortly. So Clostridium botulinum is the name of these bacteria. As you can see, they're rod-shaped. They're gram-positive bacteria. But most importantly, these bacterium, Clostridium botulinum, they make the neurotoxin botulinum toxin. And it's a protein. So thus it's going to affect your neurons. So this is C. botulinum. So this is the rod-shaped bacterium, and it makes the protein botulinum toxin. Which is a protein. Proteins are ultimately derived from what molecule? You know it. You put it on your essay. DNA. And then DNA is transcribed into single-stranded mRNA. And the mRNA can be translated eventually into this protein that can be secreted out of the cell. The botulinum toxin protein. So bacteria make proteins the same way your body and cells make proteins. Now, if we look at this botulinum toxin protein, if we look at it on the slide, notice its molecular structure is a lot more complex than a circular ball. So this is what the toxin actually looks like. And what I want you to get out of this is there's three regions associated with the protein. There is a binding domain, this part of the botulinum toxin protein, which binds to axon terminals of neurons. It's a neurotoxin, so it's going to affect your neurons and enter them. This middle part is referred to as the translocation domain, and it's going to help move this last part of the toxin into the cytosol of the neuron. So thus, this last region of the botulinum toxin protein is the catalytic domain. It's a protease that will disrupt your neurons from functioning normally. Proteases, they cut or cleave proteins. So thus, from this information, you can kind of get the idea that this toxin will enter your axon terminals of neurons, and it will cut certain proteins inside the axon terminals. And thus that will disrupt the normal functioning of those neurons. So how does this toxin get into your body? Well, C. botulinum, referring to the bacterium, it can be found in soil as well as in the ocean. So if you put your hands in the soil, or if the marine sediment is contaminated with the bacterium, and then maybe you touch your food with your hands, then the food and the bacterium can get into your body. Or the toxin can produce spores that can be found on any surface, and then those spores would enter your body. So specific mechanisms of entry, one is through ingestion, whether you ingest the bacterium itself or the spores. Sometimes if food is not canned properly or cooked properly, it can contain the bacterium or the toxin. And then you would bring it into your body via ingestion. The bacterium or toxin or spores can also enter through open wounds. This is common in drug users that inject drugs through needles. You can inhale the spores. That's another way to get the toxin into your body. Or it can also be directly injected into your body. Anyone know why we inject this toxin into our body? For cosmetic reason?
To relax the muscles.
Botox. Yeah, to relax the muscles so they don't contract and so you don't put wrinkles in your forehead. Okay, so BONT stands for the botulinum neurotoxin. Here it looks like Pac-Man. And what will happen is it will enter your cells, and then it will move through your blood, and eventually it will be uptaken by the axon terminals of your neurons. And it specifically affects axon terminals that communicate with skeletal muscles. So it specifically targets cholinergic neurons. A cholinergic neuron is a neuron that releases acetylcholine. And because it affects these cholinergic neurons, it's going to then inhibit them from releasing acetylcholine, and you won't get communication to the skeletal muscle. And if you don't get communication to the skeletal muscle, the muscle just stays in a relaxed state, which could lead to paralysis. Okay, so this is where your first potential essay question starts. It's first describing the normal communication between a neuron and a muscle at a neuromuscular junction. So we essentially covered this during our, I believe, our first nervous system lecture, but we'll review it here again. So here's your neuron, and it's going to communicate with this muscle fiber. Muscle fibers are cylindrical, and they are the cells of your skeletal muscles. They communicate using what neurotransmitter? Acetylcholine, or ACH. And I told you this term earlier. What do we call this region? When we have a neuron communicating with a muscle? A neuromuscular junction. Just as an aside, if we have two neurons communicating with each other, the area of communication is referred to as a synapse. But when we have a neuron communicating with a muscle, we change the term to neuromuscular junction. what receptors did I have on the muscle fiber that bind the acetylcholine? Who remembers? What type of receptors? We said it like 30 minutes ago. It starts with an N. Nicotinic receptors. So, on the slide, we're going to describe what happens normally at the neuromuscular junction to allow communication between the neuron and the muscle fiber to then tell it to contract. So, this is unaffected transmission or communication at a neuromuscular junction, meaning this is normal communication. So, when action potentials reach the axon terminal, what channels open? There you go. Voltage-gated calcium ion channels open. Are calcium ions higher concentration inside or outside the neuron?
Outside.
Outside, so they'll naturally rush in from high to low. Inside, calcium will bind to a sensor protein called synaptotagmin. When it binds to synaptotagmin, it forms the calcium synaptotagmin complex. And then the calcium synaptotagmin complex, it will interact with these blue snare proteins to then allow exocytosis of these neurotransmitters out into the synaptic cleft. Then those neurotransmitters can bind and activate the nicotinic receptors. And as we described 30 minutes ago, it allows for muscle contraction. So this is normal communication between a neuron and muscle, allowing for muscle contraction. Now let's draw out affected transmission at the neuromuscular junction. Meaning, what's going to happen is the toxin will enter the neuron, and it's going to prevent the release of the neurotransmitter, and that will prevent the stimulation of the muscle fiber, and thus you will not get contraction. So we're going to draw another neuromuscular junction. And I'm going to make my axon terminal really big so I can fit some things into it. Can someone hit the lights so you guys can see this better? Okay, so here is a vesicle with acetylcholine neurotransmitter, and it's docked onto the membrane of the axon terminal via snare proteins. What's the region between two cells called? The synaptic cleft. Okay. Let's draw your botulinum toxin. The botulinum toxin has a binding domain, so B for binding, a translocation domain, T for translocation, and then C for a catalytic domain, BTC. And what will happen is the binding domain of the botulinum toxin will bind to a receptor located on the axon terminal. And through receptor-mediated endocytosis, which is a type of cellular import, we will bring in the toxin into the cell. Through receptor-mediated endocytosis, which is a type of bulk transport, we bring the toxin into the cell. Specifically, we bring it into the axon terminal. Then, with help of the translocation domain, we're going to move the catalytic domain out of this vesicle. This vesicle is also referred to as an endosome. With some assistance from the translocation domain, we'll move the catalytic domain out of the endosome. What did I say the catalytic domain serves as? A protease. The catalytic domain will cut proteins, such as the snare proteins. And that will cause these vesicles to then detach and no longer be docked and ready to be released. So thus, when action potentials get to the axon terminal and calcium ions enter, and it binds to its center protein, the calcium and center protein, when it interacts with the snare protein, it can no longer allow for exocytosis of these vesicles. And thus you no longer release neurotransmitters. And if you have no ACH, you do not stimulate the nicotinic receptors of the muscle fiber, and that leads to no contraction or flaccid paralysis. So I'll describe it one more time from the top. So listen up. Are you on me?
I was just going to ask what receptor the toxin has. Like, is there specific receptors?
I don't don't know the specific name of it, but you just call it a botulinum toxin receptor. All right, you guys listening? So from the top. I heard that. Too bad you can't bring that to your exam. All right. Botulinum toxin, it affects axon terminals of your neurons. Botulinum toxin has three parts or domains: a binding domain, translocation domain, and a catalytic domain. The binding domain, it binds to a receptor located on the axon terminal. And when it binds, through a type of bulk transport we described in the first unit, receptor-mediated endocytosis, when the binding domain binds to this receptor on the axon terminal, through receptor-mediated endocytosis, the toxin is brought in to the axon terminal in a vesicle known as an endosome. Then, the translocation domain will help move the catalytic domain out into the cytosol of the axon terminal. And the catalytic domain, previously we said, it serves as a protease, meaning it cuts or digests proteins. So that the catalytic domain will then digest these snare proteins, thus detaching these vesicles that contain acetylcholine. Now they're kind of detached, they're no longer docked, and they're just floating around the neuron. Thus, when the action potential reaches the axon terminal and calcium comes in, and it makes the calcium synaptotachin complex, when that complex interacts with the snare proteins, since the vesicles containing ACH are no longer there, they can't be exocytosed and they can't be secreted. So therefore, the axon terminal does not release acetylcholine, and when you don't have it, you can't bind and activate the nicotinic receptors, and therefore that would lead to no muscle contraction. The muscle would stay relaxed, or it would be paralyzed. That's all you want? Who was Keon? Okay. Questions? No? Okay, let's talk about the fun part. So we talked about this neurotoxin, but how is it relevant to you? Targeted, meaning you only inject it to a specific region of your body, and low dosage injections of the botulinum toxin are used for cosmetic and clinical purposes, meaning you don't inject a high concentration of it into your body, because if you do, you might cause major paralysis, which you have. So, just in this diagram right here, we have 3 neurons that usually release acetylcholine onto these different cells. So let's go through it. You can use botulinum toxin, aka Botox, and inject it into the forehead or around the eyes to stop communication to your facial skeletal muscles. And when you tell those muscles not to contract due to the toxin, it prevents creases in the skin and it reduces or prevents wrinkles. So that's the clinical application, or not clinical, cosmetic application of that. Clinical treatments, one could be to reduce muscle spasms. Those are involuntary contractions of your muscles, and sometimes they can be painful. So thus, in that region where you're experiencing the muscle spasm, whether it's your back, your legs, you inject botulinum toxin there to prevent communication to those muscles so that they relax and so that they do not spasm. In the urinary bladder, sometimes you can have an overactive skeletal muscle that contracts too frequently, and that will push the urine out, leading to more frequent urination. Or urinary incontinence, where you can't control when you want to pee. So thus, to relax that overactive muscle in the urinary bladder, you can inject botulinum toxin there to prevent the contraction of it. Strabismus is when the muscles that control the position of your eyes are sometimes overactive. So you may be cross-eyed, or maybe one of your eyes may point a certain direction. So to correct that, you may inject the toxin into certain eye muscles to realign the positioning of your eyes. You'll see in the second part of the image that neurons can release acetylcholine onto sweat glands, leading to perspiration. For some individuals, or more specifically models who don't want to sweat and ruin the expensive clothing, you can inject botulinum toxin to reduce sweating. And for individuals that suffer from migraines, those neurons that signal pain for the migraines, if you inject botulinum toxin, you can reduce the communication between those neurons to reduce the pain transmission of migraines. So those are some clinical uses of the toxin to treat certain conditions of the body. Or you can also use it cosmetically. So if you inject it in low dosages and at a specific part of the region, it can have many beneficial purposes. Sound good? Let's do today's lab, which is on vision.